Hello. So today we'll be discussing erythropoiesis and structure and function of red blood cells. So by way of introduction, erythropoiesis simply describes the process of the origin, development, and maturation of erythrocytes. That's red blood cells. All right. Now, in humans, erythropoiesis occurs almost exclusively in the red bone marrow. Right? There's other organs that can produce red blood cells in special situations. I will talk about them later. Now, the red bone marrow of virtually all bones produce red blood cells from birth up to about 5 years of age. And between the ages of 5 to 20, the long bones they slowly lose their ability to produce red blood cells. Above the age of 20, most red blood cells are produced primarily in the marrow of the vertebrae, the sternum, the ribs, and the pelvis. You'll notice that this is essentially a list of flat bones. Now, in utero, from around the third week of intrauterine life, we have the mesoblastic stage where when nucleated red blood cells form in the yolk sac and also in the placenta. Then we have the hepatic stage from around the sixth week onwards. Red blood cells are formed in the liver, spleen, and lymph nodes. Then we have the myeloid stage where the bone marrows gradually become the principal source of red blood cells. Now, after birth, up to 5 years, the bone marrow of all bones produce erythrocytes. So, but between 5 to 20 or to 25 years, the red marrow uh, of all bones only. Then, after 20 to 25 years, the red blood cells are produced mainly in membranous bones. Uh, some examples are the flat bones we've mentioned earlier, as well as the ends of the long bones. And after what happens is that after the 20 years of age, the shaft of the long bones, the red marrow inside the shaft gradually turns yellow. The red marrow turns to yellow marrow because of fat deposition. It thus loses its ability to produce um, red blood cells. Now, in adults, the liver and spleen may sometimes produce red blood cells if the red marrow is destroyed or fibrosed. Um, Now, we know that the bone marrow is the site of production of all blood cells, but most of the bone marrow is actually involved. That's about three quarters, 75% of them is involved in production of white blood cells, and only a quarter is involved in production of erythrocytes. Okay, so this shows the normal bone marrow conversion, right? In this case, a long bone. At birth, virtually the whole marrow, all of the marrow in the long bone produces red blood cells. But over time, by adulthood, it's only the ends of the long bones that produces erythrocytes. Right now, this um, this lecture is about erythropoiesis. So erythropoiesis is just one of is just a part of hematopoiesis. Hematopoiesis is the production of blood cells, right? Whereas erythropoiesis is the production of red blood cells. Okay, so all right. So now this whole schematic shows hematopoiesis, but what we are interested in, this is the part we are interested in here. This is the red cell uh, part. You can see the red cell lineage here. All right. So this is a similar schematic. All right. We can also see something similar here. Right. The production of all blood cells starts from the multipotent hematopoietic stem cell, or also referred to as a pluripotent hematopoietic stem cell. All right. Which first differentiates into the lymphoid and myeloid lineages, all right, which now further differentiates into specific progenitor cells, all right. So here, this gives rise to the granulocyte monocytes progenitor, which we in turn give rise to myeloblasts, which eventually leads to formation of neutrophils. Now the monoblast eventually forms monocytes, and so on. All right. So, but for since we are interested in erythropoiesis here, this is the part we are concerned with. This particular progenitor cell could give rise to either a red cell lineage or platelets, megakaryocytes, right, which eventually lead to platelets. All right. So, all right, let's move. Okay. Now, let's talk about the theories of erythropoiesis. Now, the major theory that is accepted by most people, most authorities, is referred to as the monophyletic theory, which holds that all cells arise from a single common stem cell. Right? There's also the diphyletic theory, which holds that lymphocytes and monocytes are derived from one specific stem cell, whereas granular leukocytes and red blood cells from another stem cell, referred to as myeloblast. Then there is the polyphyletic theory, which holds that a separate stem cell exists for each blood type. All right. So, but like I said, it's the monophyletic theory that is most widely accepted. Okay. Now, the um, yeah. So the blood cells, differentiating blood cells can be grouped into three general categories. We have the pluripotent stem cells, then restricted progenitor cells, then functional blood cells. So the pluripotent stem cells is that cell, right? That has the capacity to give rise to several different lines of specialized cell. Hence the term pluripotent. We say that cell is pluripotent, or it could also be referred to as a multipotent. Okay, so and in these stem cells, the nucleus is undifferentiated. Basically, under the microscope, it will look like a large lymphocyte, right? It's undifferentiated. Okay, the cytoplasm is basophilic because of the presence of ribosomes. Basophilic cytoplasm just means that the cytoplasm stains with basic dyes as opposed to acidic dyes. All right, and these pluripotent stem cells are capable of extensive proliferation by mitosis. Okay, then we have the restricted progenitor cells. Um, as we move from the pluripotent stem cells to these restricted progenitor cells, the the direction in which these cells will differentiate, where they will end up in the process of differentiation, increasingly becomes restricted. Firstly to either myeloid or the lymphoid, right, and finally to the progenitor cells specific for a certain blood cell. All right. So at this point, they are more differentiated than the pluripotent hematopoietic stem cell, but they are still relatively undifferentiated because, for instance, the myeloid uh, progenitor cells could still either give rise to the lineage that would that would lead to formation of a red blood cell, or it could lead to formation of a platelet. So at that point, right, the myeloid progenitor cell is still, it has differentiated a little, right, because it has clearly separated from the lymphoid lineage that will give rise to lymphocytes and monocytes, but it still has possible it still has possible options in what it will eventually end up as. Okay. Now, proliferation and differentiation of these restricted progenitor cells is under control of several factors that are specific to each cell lineage. If you go back to this, all right, so we can see some of these factors mentioned here. For instance, this particular uh, this particular lineage that gives rise to red blood cell. At this point, it's under the influence of several factors, including granulocyte macrophage colony stimulating factor, right? Here, granulocyte monophage colony stimulating factor, as well as thrombopoietin, gives rise to or platelets. Now, here, this erythro stands for erythropoietin, right? So, erythropoietin is involved in the formation of red blood cells. Thrombopoietin is involved in the formation of platelets and so on. And even some interleukins. Interleukin 5 is here, acts, helps regulate the formation of eosinophils. Interleukin 4 and 3, basophils, and so on. Okay. So, eventually, with uh, recurrent differentiation, the progenitor cells become recognizable, and they are become recognizable. You know, the lineage, uh, the mature cells. We are able to eventually see the matured cells, and at this stage, they are termed blood cell precursors, which then mature into functional blood cells. Okay. Now, erythrocytes. Let's talk about the process of erythropoiesis. Erythrocytes arise initially from the uncommitted pluripotent hemopoietic stem cell (PHSC) in the bone marrow, right? And the this pluripotent cell divides and differentiates to give rise to committed pluripotent stem cells. Now, remember what we said earlier, right? That the initial stem cell that could give rise to any blood cell type is the uncommitted pluripotential hematopoietic stem cell. So, but when it differentiates and forms the myeloid progenitor cell. Now, that myeloid progenitor cell could still either give rise to either red blood cells, mature red blood cells, or platelets. All right. So at that point, we say that it's still a stem cell, right? But it has committed itself to the myeloid lineage, right? As opposed to committing itself to the lymphoid lineage. All right. So, and of course, as these cells reproduce, they reproduce and differentiate, but a small portion of them remain exactly like the original cells, right? So that at all times we always have some pluripotent hematopoietic stem cells within the bone marrow to maintain their supply. Okay. All right. So the uncommitted stem cell could give rise, yes, as I've said, to either myeloid stem cells or lymphoid stem cells. Now, myeloid stem cells, also known as colony-forming blastocytes. Now, this give rise to different units of colony-forming cells. So, colony-forming blastocytes will give, in turn, differentiate to form colony-forming unit erythrocytes, CFUE, along with others, right? Colony-forming unit granulocyte, monocytes, colony-forming unit megakaryocyte. All right. So, but this is the one we are interested in since this lecture is about erythropoiesis. All right. So, if we are talking about the process of erythropoiesis, it starts with it starts with the uncommitted pluripotent hematopoietic stem cell, right? Then it now leads to the committed pluripotent hematopoietic stem cells, which will differentiate into either myeloid or lymphoid. Now, for erythropoiesis, it will be myeloid stem cell, aka colony forming blastocyte. Then the forming blastocytes now differentiate into several cells, but the ones we are interested in will be the colony forming unit erythrocytes, CFUE. Okay, so it is this colony forming unit erythrocytes that will eventually lead to uh, the formation of blood cells, of red blood cells. Okay, so now the we should note that the growth and reproduction of different stem cells are controlled by multiple proteins, which we refer to as growth inducers. Interleukin 3 is one of these, which helps, which promotes the growth and reproduction of virtually all the different types of committed stem cells. All right. So, but the others induce growth of only specific type of cells. Then we also have differentiation inducers, right, which induce differentiation of committed stem cells uh, towards a final adult blood cells. Now, some of the things that happen, right, during the process of growth and differentiation, or from the stem cells to the formation of the mature red blood cells. Some of the events that happen is that there is a reduction in size from about 25 micrometer in diameter to 7.2 micrometer of the matured red cell. Right, so the stem cells, the initial stem cells are about this big, but by the time the red blood cell, the mature red blood cell is formed, it's just about a little over 7 micrometer in diameter. Then there is condensation and eventual loss of the nuclear material and the organelles. Right, mature red blood cells do not have nucleus, do not have mitochondria, and some other organelles. Now, there is acquisition of hemoglobin via protein synthesis. There's also loss of the proliferative capabilities. Mature red blood cells cannot reproduce; they cannot reproduce themselves. Right? Then there's a change in the staining properties of the cytoplasm. Now, erythropoiesis is can be divided into an early and a late stage. Now, the early stage is characterized by proliferation of early stage erythroid cells, which we refer to as progenitors, and the late stage erythropoiesis involves differentiation and maturation of late stage erythroid cells, referred to as precursors. This was shown in one of the previous previous schematics. Let's go back a bit. Okay, so here we can see now. I'm looking for the yes. This is the erythrocyte lineage. All right. So at this point, these are progenitor cells, right? But from the pronormoblast, or also known as the proerythroblast, downwards, from the pronormoblast or proerythroblast down to the reticulocytes, they are referred to as precursor cells. But everything between this and the original uncommitted stem cell, everything here, they are referred to as progenitor. So this is the initial stem cell, then we have the progenitor cells, then we have the precursor cells, finally we have the matured red blood cell. Okay. All right. So let's now talk about stages in of still talking about stages of erythropoiesis. Now, you should know that if you were to write on the stages of erythropoiesis, you would want to start with uh, from the initial uncommitted pluripotent hematopoietic stem cell before. Okay, so you want to start. Okay, let's go back a few slides. If you are to write on erythropoiesis, yes, you want to start by mentioning the uncommitted pluripotent hematopoietic stem cell, right? Followed by the committed hematopoietic stem cell, which for red blood cells will be the colony forming blastocytes. Now, the colony forming blastocytes will in turn differentiate to give us the colony forming unit erythrocyte. Now, it is this particular cell that will now differentiate to give us the proerythroblast. So before the proerythroblast, we've had three levels of differentiation, right? There was the initial uncommitted stem cell, which differentiated into these committed stem cells, right? Which called the colony forming blastocytes, which then differentiated into the colony forming unit erythrocyte, which then differentiated again to give us the pronormoblast, also called the proerythroblast. All right. So it's after this stage that you will now start talking about the proerythroblast up until the matured red blood cell. Okay, so let's go back there now. So, all right. So the proerythroblasts, all right. It's also referred to as the megaloblast. It's a large cell, right? The uh, 20 to 30 micrometer in diameter. It has a large spherical nucleus. It has two or more nucleoli. The cytoplasm is basophilic. It has high nuclear to cytoplasmic ratio. Now, what does this mean? It means that the ratio of the okay, when we look at the cell under the microscope, right? When we say a cell has a high nuclear to cytoplasmic ratio, it simply means that the nucleus virtually fills the whole picture of the cell that we see. That there's usually just a thin rim of cytoplasm. All right. So now in this stage, synthesis of hemoglobin starts, but the hemoglobin is not yet visible, right? But the synthesis has actually started. Now, so it divides and differentiates further to lead to the formation of the basophilic erythroblast, right? Now, this cell is still large, right? About this 15 to 20 micrometer in diameter. It still has a high nuclear to cytoplasmic ratio, but it has no nucleoli, right? As a you know, compared with the proerythroblast that has two or more nuclei. So the basophilic erythroblast has no nuclei. It has a basophilic cytoplasm, and the chromatin condenses. Right. So, but again, as I said earlier, basophilic cytoplasm just means that the cytoplasm stains with basic dyes. They are basic dyes, they are acidic dyes, right? So basic dyes stain both the proerythroblast and the basophilic erythroblast. All right. So the next stage is the polychromatophilic or polychromatic erythroblast, also referred to as intermediate normoblast. Yes, this size has reduced a bit more, and hemoglobin starts to appear. It starts to become visible under the microscope. The cytoplasm is still basophilic, right, and appears blue because of the presence of ribosomes. But there are also some parts of the cytoplasm that appear pink, right? So and that's so yeah, so at the same time it appears as if the cytoplasm appears blue and pink, basophilic and acidophilic, hence the name polychromatophilic, right? That it has more than one color. That's referring to the cytoplasm. All right. So the cytoplasm has diffuse basophilic and acidophilic staining. The nucleus is still present, spherical, and chromatin has gotten even more condensed. Now, the first three stages up until this polychromatophilic erythroblast, they are capable of proliferation, right? That they can still reproduce themselves, right? So the proerythroblast, the basophilic erythroblast, and the polychromatophilic erythroblast can proliferate. They can reproduce themselves. Now, after this, the next stage is the autochromatic erythroblast. The size of the cell is getting smaller. The nucleus is still present, but it's very small and dark staining. Uh, all right, the quantity of the hemoglobin increases. The cytoplasm becomes almost acidophilic, meaning that the pink staining, whereas in the polychromatophilic erythroblast, we said that the cytoplasm was had diffuse blue or pink staining, right? Basophilic and acidophilic respectively. For the next stage, the acidophilic staining is beginning to predominate. So it's almost completely acidophilic, almost completely pinkish, but there will still be a bluish tinge to it. So before it moves on to the next stage, the nucleus disintegrates and disappears, a process referred to as pycnosis, and its remnant is extruded from the cell. All right. So the next stage after this is now the reticulocyte. Reticulocyte. All right. So it has no nucleus. The nucleus has been. So now take note that the nucleus is extruded, not in the reticulocyte stage, but in the stage prior to the reticulocyte. Right. All right. So the reticulocyte has no nucleus. The cytoplasm becomes even more acidophilic, right? But there is still some basophilia because there are still remnants of some organelles, right, such as the mitochondria and Golgi apparatus. All right. Now the cytoplasm, uh, the cell is called the reticulocyte because the cytoplasm still contains the reticular network, like a kind of net, like a kind of net-like structure. All right, formed from the remnants of disintegrated organelles. That's why it's called reticulocyte. All right, so some reticulocytes will actually enter the circulation along with mature red blood cells via the process called diapedesis. Okay, and so in peripheral blood, in neonates, you might have as many as 2 to 6% of reticulocytes in peripheral blood, but in adults, in an normal adult in normal health, it's expected that reticulocytes count in peripheral blood should be 1% or less. Okay, so this is the process of diapedesis. We can see this is inside of the bone marrow. The stem cell eventually leading to formation of reticulocytes. You can see the reticulocyte expelling the nucleus and the mature red cell, or and some reticulocytes to squeeze through the endothelium, the pores between the endothelial cells, and enters uh, into the circulation. All right. So finally, the mature erythrocytes. The reticular network disappears. The cell attains the biconcave shape, which we know it for, and decreases in size. The cytoplasm now stains completely acidophilic, so it's completely pink when we look at it under the microscope. All right. So now this process uh, of differentiation and maturation from the proerythroblast to the mature red blood cells takes about 5 days, about 7 days. I mean, 5 days from proerythroblast to reticulocytes, and about 2 days for the reticulocytes to mature to become to be for the reticulocyte to become the matured red blood cell. All right, now let's talk about erythrokinetics. Um, yes, about 1% of red blood cells are removed and replaced daily, and the uh, the process is regulated by to a large extent by hypoxia, right? When the kidney senses hypoxia, okay, and on average, erythrocytes circulate for about 120 days, and they are removed by macrophages in the spleen, not just the spleen, bone marrow, and other parts of the body referred to as the reticuloendothelial system. All right, once they are no longer viable. All right, erythropoiesis is stimulated by erythropoietin, which is a hormone produced by the kidney. We'll talk more about erythropoietin later. All right, secretion of erythropoietin is stimulated by hypoxia. All right, hypoxia is just a situation where there is a low oxygen tension in tissues, and it can be caused by anemia, hemorrhage, high altitude, lung disease, heart failure, and some other conditions. All right, when the kidney senses hypoxia, right, when the amount of oxygen getting to the kidneys, that's hypoxia. When it's low, hypoxia, there is a substance called hypoxia inducible factor activates the gene responsible for production of erythropoietin. Okay, and so this increases erythropoietin synthesis. Okay, erythropoietin stimulates erythrocyte formation in the bone marrow. Okay, and so and subsequently, when the increased red blood cell formation leads to increased oxygen delivery to the tissue, uh, this eventually leads to a situation, right, where the the erythropoietin production synthesis is now down regulated. All right. Now, there are some factors that are necessary for erythropoiesis. Okay, there are general factors, maturation factors, and factors that are necessary for hemoglobin formation. Now, some general factors include things like hormones like erythropoietin, thyroxine, and the various growth factors, some vitamins, and so on. Now, let's start with erythropoietin. Okay, it's a glycoprotein with 165 amino acids, um, mainly secreted by peritubular capillaries in the renal cortex in response to tissue hypoxia. Okay, uh, some erythropoietin is secreted by the liver and brain. Now, renal tissue hypoxia leads to increased tissue levels of hypoxia inducible factor, I mentioned earlier, which and this factor ultimately leads to increased erythropoietin synthesis. Okay. Now, hypoxia in other parts of the body can also stimulate erythropoietin production. Okay, and this suggests that They are probably non-renal sensors that can send signals to the kidneys. All right. So epinephrine, norepinephrine, several prostaglandins can also stimulate erythropoietin production. And conditions like anemia, high altitude heart failure, hemorrhage, lung disease, which we mentioned earlier, would also increase erythropoietin production. Now, what does erythropoietin do? It stimulates production of pro-erythroblasts, right, and thus of erythroblasts, right. So it is from pro-erythroblasts, right, downwards, along the lineage, right. Erythropoietin will exert its effect on pro-erythroblasts and erythroblasts. So it will not affect the growth or differentiation of stem cells, right, of either the original pluripotent, uncommitted pluripotent stem cell or the committed stem cell. No, it will start working at the level of the proerythroblast downwards. All right. So it will cause them to increase their cell division rates and differentiation, and also cause them to the proerythroblast to pass more rapidly through the different erythroblastic stages than they usually do. All right, and it also accelerates the rate of hemoglobin synthesis. Okay. All right. Now the okay. So in the absence of erythropoietin, few red blood cells are formed by the bone marrow. So erythropoietin is absolutely essential. Right. So people with renal failure, with kidney failure, who are unable to produce erythropoietin, they generally have chronic anemia, right? And they often need to have synthetic erythropoietin given. All right. So when the when now it's possible when for the rate of red blood cell production to increase to more than 10 times the normal rate if the factors necessary are present. Erythropoietin, iron, and the required nutrients. If they are all available, then the rate can go as high as more than 10 times normal. Now, thyroxine, thyroid hormone, also accelerates erythropoiesis. Then we have hematopoietic growth factors, the various interleukins, interleukin 3, 6, 11, and others. Then vitamins, right? Deficiency of vitamins, of particular vitamins will cause anemia. So all of these vitamins B, C, D, E, they are important for erythropoiesis. Then we have maturation factors. Yeah, vitamin B12 specifically, intrinsic factor, folic acid. They are required for maturation of erythrocytes. Okay. Now, vitamin B12 and folic acid are especially important because they are essential for synthesis of DNA. They are essential for synthesis of DNA. All right, so let's start with vitamin B12. It's also referred to as extrinsic factor, uh, and is obtained almost exclusively from diet. Um, its absorption from intestine requires the presence of the is a substance called referred to as the intrinsic factor of Cas2. All right. The okay, some vitamin B12 can also be produced by intestinal flora in the large intestine. Okay, and it is stored in the liver, uh, mostly in the liver and in muscle, and released slowly as needed to the bone marrow. Okay, so as I said earlier, it's essential for synthesis of DNA in red blood cells. Okay, all right. So the minimum amount of vitamin B12 required each day uh, to maintain normal red blood cell maturation is only about 1 to 3 micrograms. All right, and the normal storage of vitamin B12 in the liver and other tissues is about 1,000 times this amount. Right, so for daily red blood cell production, the bone marrows need they need only the bone marrows need only 1 to 3 micrograms, but we have more than 1,000 times this amount stored in the liver and other tissues. And so what this means is that it will require 3 to 4 years of a problem with vitamin B12 absorption, defective vitamin B12 absorption. It will require 3 to 4 years of this uh, uh, before anemia forms, maturation failure anemia. We said that vitamin B12 is required for maturation of red blood cells. So before anemia due to maturation failure from vitamin B12 deficiency, it will require 3 to 4 years of this before the anemia sets in. Okay, so deficiency of vitamin B12 leads to failure in the nuclear and cell division. All right, remember that again. We said vitamin B12 is involved in DNA replication. So, deficiency of it will cause failure of nuclear and cell division. Okay, and in these instances, the cells, the erythroblastic cells, will produce larger than normal red blood cells called macrocytes. Right, so anemia due to vitamin B12 deficiency, will lead to formation of larger than normal red blood cells called microcytes. So it's called a macrocytic anemia. Okay, microcytes have a fragile membrane. They are irregular, large, and oval instead of the usual biconcave disc. They usually have a short half-life. Okay, vitamin B12 deficiency causes pernicious anemia, which is a macrocytic anemia. It's also a megaloblastic anemia. Alright, megaloblastic anemia is a subset of macrocytic anemia. So there are other types of macrocytic anemias that are not megaloblastic. Alright, just take note. So megaloblastic and macrocytic are not the same thing. Megaloblastic is a subset of macrocytic anemias. Alright, so vitamin B12 is also referred to as anti-pernicious factor, and thus its deficiency, the anemia caused by deficiency, is also referred to as pernicious anemia. Now, the intrinsic factor of capsule that we mentioned earlier, this is produced by parietal cells of the gastric glands in the gastric mucosa, and its importance is that it helps in the absorption of vitamin B12 from the intestine. Okay, so its deficiency will also lead to pernicious anemia, since its deficiency will lead uh, to poor absorption of vitamin B12, which will then lead to vitamin B12 deficiency, hence leading to pernicious anemia. All right. So theoretically there is a substance called Martinic principle, which is thought to be produced by the action of intrinsic factor on extrinsic factor. It's referred to as the anti-anemia principle as well. Now, folic acid, I believe, is a vitamin B9. All right, it's also essential for the synthesis of DNA. Okay, so its deficiency will also lead to formation of megaloblastic anemia. Now, factors that are necessary for hemoglobin formation. Yes, of course, we have the first class protein. Hemoglobin is a protein. All right, so we need first class protein amino acid. First class proteins are proteins that contain all the nine essential amino acids. All right, the nine essential amino acids. All right, so the first class proteins, amino acids, iron for the formation of the heme part of hemoglobin, copper for absorption of iron from the GIT, cobalt and nickel, and these other vitamins, they are also necessary for uh, hemoglobin formation. Now, there are other hormones that are involved in that influence erythropoiesis. Thyroid hormone we mentioned earlier, androgens, right? Testosterone, cortisol, growth hormones. Uh, Deficiency of any of these also results in anemia. All right, so let's talk about ineffective erythropoiesis. So sometimes there are situations uh, um, in which erythropoiesis, the process of erythropoiesis leads to the formation of erythroid precursor cells that are defective, right? These defective cells will then usually die by apoptosis in the bone marrow before they make it to the reticulocyte stage. All right, This is effective erythropoiesis. So it is defined by expansion of the early stage. So if we were to take a bone marrow biopsy and examine it under the microscope in conditions that cause ineffective erythropoiesis, we would see that uh, there are a lot of the early stage erythroid precursors, right? The early stage, such as the pro erythroblast, for instance, that's an example, right? So you might see a lot of pro erythroblasts, a lot of um, uh, uh, erythroblasts, and so on, right? But they will not mature. So they will, the numbers of the pro erythroblasts will expand beyond normal, right? When we look at the bone marrow, when you look at the bone marrow under the microscope, but they will not progress to the point where they form reticulocytes and mature red blood cells. They would, a lot of them will die, right, by apoptosis, and so you end up having so many of these immature erythroid cells in the bone marrow. But when you take the peripheral blood, when you take blood sample from the circulation, you find out that the blood is anemic because a lot of these cells have died due to apoptosis. All right. So several conditions can lead to ineffective erythropoiesis, such as the megaloblastic anemia, which we've discussed earlier. Right. So megaloblastic anemia from vitamin B12 or folic acid deficiency, which will lead to failure of DNA synthesis. Right. So many of these cells will die before they get to the reticulocyte stage or the mature red blood cells. They will die by apoptosis. Then thalassemias, sideroblastic anemias also will cause ineffective erythropoiesis. Okay. So now, what now happens? So because of the anemia, right? Because of the anemia that is thus caused, right, will lead to low oxygen tension in the tissues. It will lead to hypoxia. Yeah, lead to hypoxia, and this hypoxia will now trigger the kidneys to increase erythropoietin production. All right. And the erythropoietin production will, again, what did we say that the erythropoietin does? It acts at the level of the pro-erythroblast to increase the growth and differentiation of this, and so all the precursor cells from the pro-erythroblasts downward, yes, these precursor cells, they will increase in number, right? So it will now turn into a kind of vicious cycle. They will increase in number, but because as long as the condition causing the ineffective erythropoiesis is still ongoing, then this increased pro-erythroblasts and erythroblasts, they will still die by apoptosis. Many of them will still die by apoptosis before they become reticulocytes, and so it just goes on and on. The anemia persists until the cause is treated. Now, there's also a condition referred to as insufficient erythropoiesis. This is just a decrease in number of erythroid precursors in the bone marrow, resulting in decreased red blood cell production and anemia. Okay, so as opposed to ineffective erythropoiesis, where the erythroid precursors were actually expanded more than normal, like the numbers were far more than normal, but many of them just died before they matured. In the case of insufficient erythropoiesis, the problem is that the precursors are very few, right? So those few precursors will lead to um, to the development of few matured red blood cells. All right, so things like iron deficiency can cause insufficient erythropoiesis. Deficiency of erythropoietin, such as people with kidney failure, renal disease, or uh, loss of the erythroid precursors as a result of autoimmune reaction, as in aplastic anemia, uh, or due to infection such as parvovirus B19 infection, or the bone marrow itself might be invaded, might be infiltrated with cancerous, with leukemia cells, right? Well, which will prevent the bone marrow from carrying out erythropoiesis as it should. All right. So let's now talk about the morphology of erythrocytes. Yes, we've referred to this earlier. Mature red blood cells have no nucleus, they have no cell organelles, they have no granules, unlike some of our white blood cells. Mature red blood cells have a biconcave shape, right? They have a biconcave shape, which leads to a relatively large surface area to volume ratio, right? The surface area compared to the volume, like is quite large, and this helps to allow rapid gas exchange and movement of substances across the membrane. This biconcave shape also helps to provide great flexibility because red blood cells can pass through uh, tiny blood vessels, through capillaries that have a diameter that is lesser than the diameter of red blood cells. So this biconcave shape allows uh, the red blood cells to deform their shape as they are passing through these tiny capillaries, and then they now form the shape back after they pass through. Now the mean diameter is about 7.8 micrometer. Thickness is about 2.5 micrometer at the periphery and less than 1 micrometer in the center. All right. So this is a red blood cells, a micrograph of red blood cells, clearly showing their biconcave shape. All right. Okay. So this also show the biconcave red blood cells. So the red blood cell is simply a bag of fluid, right? And the bag is shaped a bit like a donut. That's the biconcave shape, right? With dissolved substances and hemoglobin inside. The surface of the cell is negatively charged. The membrane has an outer glycoprotein coat. It has a lipid bilayer, like many other cells. Uh, it has its inner cytoprotein cytoskeleton with proteins such as pectin, actin, ankyrin, and so on. The surface area usually about 120 micrometer square, volume about 85 to 90 micrometer cube. The mature red blood cell does not have mitochondria, so it produces energy by glycolysis and by the pentose phosphate pathway. It does not have insulin receptors, so glucose uptake is not insulin dependent. All right, and each red blood cell contains in health the healthy red blood cell contains about 29 picogram of hemoglobin. Uh, the uh, the shape of the red blood cell can change remarkably as the cells squeeze through capillaries. All right, okay. So now you should note that the total red blood cell surface area equals to about 2,000 times the total surface area of the body. What are we talking about? We're saying all the red blood cells inside our bodies, right? The surface area of each of them, uh, the many millions or billions of red blood cells in us, in our bodies, the surface areas of each of them combined is about 2,000 times the total surface area of the body. All right. So this huge surface area allows uh, for greater gaseous exchange, oxygen and carbon dioxide, and passage of substances in and out of red blood cells. Right. Then the shape also lends itself to the formation of stacks. Right. The red blood cells can stack on each other as if you are stacking coins. As if you are stacking coins one on top of each other. All right. So this allows them to stack themselves and facilitates transport in small vessels. All right. And as we said again, the membrane of the red blood cells is also flexible and allows them to deform and pass through very narrow capillaries. Okay, so you can see an example of rouleau formation, stacks of red blood cells. That the red blood cells are trying to pass through a narrow capillary, which is all right. So they are stacked in this way to make their passage more efficient. All right, now let's talk about hemoglobin. Each hemoglobin molecule has two alpha and two beta chains. Uh, The protein is similar to myoglobin in muscle cells. Each chain has a single heme molecule. All right. Each heme contains an ion that interacts with oxygen to form oxyhemoglobin. All right. Now the oxygen binding is of course reversible. Right. Red blood cell has about 80 million hemoglobin molecules, and each hemoglobin molecule has four heme units. All right, has four heme units, and so it means that um, each so that means that each red blood cell can bind. Just multiply this 280 by 4. It means that each red blood cell can carry more than 1 billion oxygen molecules. All right. So we should note that most of the oxygen in blood is bound to hemoglobin, about 98.5. So about it's only about 1.5% that is dissolved in plasma. Now let's talk about the fate of red blood cells. What happens to red blood cells when they get to the end of their life? Now, we said earlier that the lifespan is about 110 to 120 days, and most 90% of senescent just means old red blood cells. So 90% of these are captured and destroyed in the reticuloendothelial systems, which includes the spleen, the bone marrow, the liver as well. All right. So, but about 10% of them will hemolyze. Right? They would hemolyze spontaneously. Hemolyze meaning that they burst. Essentially, they burst spontaneously while they are moving through the blood vessels. All right. So now the senescent cells are identified by cells of the reticuloendothelial systems because they lose their deformability. All right. So while these cells are trying to pass through the capillaries of the organs of the reticuloendothelial system, right? The Because these cells are old, right? The membranes have lost their deformability. All right. So as they are passing through these tiny red blood cell, these tiny capillaries, since they are not able to deform, they will actually be destroyed, right? If they cannot, because the membranes will be non-compliant, they can't squeeze and deform and then reform after passing through. They will just get they would lyse while trying to pass through these tiny blood vessels. And then senescent cells also express abnormal proteins on their surface, right? These abnormal proteins will be recognized by macrophages of the reticuloendothelial system, and so the macrophages will know that this any red blood cell expressing these particular proteins is senescent, and it is due to is for destruction and processing. Okay, uh, they are also coated in autologous anti antiband 3 IgG, which is referred to as opsonine. So they are opsonized. Then there's reversal of membrane phosphatidyl serine. Okay, so the most of the destruction of this senescent red blood cells happens in the red pulp of the spleen. All right. Okay. So let's talk about the events that happen within the macrophage. So basically, what happens within the reticular endothelial system? Okay, using the spleen as an example, is that the cells will be identified. Some of them will burst while they are trying to squeeze through the capillaries of the red pulp of the spleen, or some of them might will be identified by macrophages in the spleen, due to expressing some of the expressing some proteins on their membrane. All right. So what happens? The macrophages will now engulf. They will phagocytose either the remnants of the red blood cells that have been destroyed, or the ones that have expressed senescent factors. So they will phagocytose them, right? They engulf old red blood cells before they rupture. Now, so inside the macrophages, the hemoglobin is degraded into iron, globin, and porphyrin. The iron combines with a protein called apoferritin to form ferritin, which is a storage form of iron. Now, the ion may enter into the blood and bind to transferrin. Transferrin is a transport form of ion. All right. So if it can bind to transferrin for transport to other parts of the body. All right. So this schematic shows what happens within the macrophages. So after you can see, okay, this is a red blood cell, right? Reticulocytes, red blood cells. Then when they are towards the end of their life, about 10% of them will lie spontaneously. 90% of the other 90% will be removed from circulation within the reticuloendothelial system. All right, so within the reticuloendothelial system, the macrophage will phagocytose these senescent red blood cells. All right, and the components will be broken down. All right, the heme will form biliverdin and eventually bilirubin. Okay, so the globin will be disassembled. Globin is a protein, right? To be disassembled, and the constituent amino acids will be recycled and used by the body. Now the heme is broken down to first to biliverdin, which is a greenish pigment, then to bilirubin. So the bilirubin now moves out of the macrophage into the bloodstream and moves to the liver, where it is excreted in bile, right? And any hemoglobin that is not phagocytosed, right? So, for instance, there will be some of the 10% of old red blood cells within the that lies within the circulation, and some of this will end up not being phagocytosed by macrophages. So this will eventually break down into its protein chains, and they will be excreted in urine. Okay, and in conditions where an abnormally large number of red blood cells lies within the circulation, it can lead to passage of brownish red urine, a condition called hemoglobinuria. When two when a lot of when there is a lot of intravascular lysis, right? So the hemoglobin will be excreted. Excess hemoglobin in the blood will be in excess free hemoglobin, right? That is circulating in blood and not within red blood cells. They will be excreted in they will break down and be excreted in urine, and they will give a brownish, a dark brown coloration to urine. Now, this urine will be as dark as Coke, as the soda drink Coca-Cola. Now, in the liver, what happens in the liver? Now, the bilirubin that, if you come back to the events in the macrophages, we said that the heme was broken down to biliverdin, then converted to bilirubin. Then the bilirubin enters into the bloodstream and goes to the liver. All right. So what happens in the liver to the bilirubin? So the bilirubin is excreted in bile. All right. If the bile ducts are blocked, or if there's any other condition that affects the ability of the liver to process bilirubin, this will lead to increase in the blood bilirubin levels, right? And in this case, the bilirubin will diffuse into peripheral tissues, right? And it can, and this causes the yellow color in skin and eyes that is referred to as jaundice. All right. So if there's any issue with the liver and the liver cannot process bilirubin properly, right? You know that every second red blood cells are dying and bilirubin is being produced. My macrophages are phagocytosing the old cells, and bilirubin is being released from these macrophages into the blood. So if there's any problem with the liver, or if the amount of blood of bilirubin is being produced is more than what the liver can handle, eventually the bilirubin will leak, will diffuse into the tissue and turn in people who are fair colored, we are fair skinned, or in people with Caucasians, for instance, yes, we actually see a yellowish tinge to their skin. Or in dark colored people, it's more noticeable in the eyes, right? The eye, there's a yellowish discoloration to the sclera, which we refer to as jaundice. Now, in the large intestine, bilirubin, we said that bilirubin is excreted in the bile. If you come back to here, bilirubin is excreted in bile, right? Now, and bile is secreted from the gallbladder into the intestines when we eat, right? Especially fatty food. All right. So the bilirubin within the bile, right, is converted to urobilins by bacteria, converted to urobilins and stachybilins, and these are excreted in feces, giving feces its normal yellow brown or brownish coloration. Okay, so these are events occurring in the liver. Alright. Now let's talk about functions of red blood cells. Yes, the chief function of red blood cells is to transport oxygen from lungs to tissues. Right? Another function is transport of carbon dioxide from tissue to lung. Okay. Um, so hemoglobin combines with carbon dioxide to form carb hemoglobin, which transports about 30% of carbon dioxide. Okay, and there's also formation of bicarbonate catalyzed by carbonic anhydrase. All right. So the uh, so about around 63% of carbon dioxide is transported to the lung as bicarbonate, then converted back to carbon dioxide when they get to the lungs. All right. Another function of red blood cells is they are important in blood group determination because blood group antigens are present on the membranes of red blood cells. Then, if another important function of red blood cells is due to the acid-base buffering that the hemoglobin participates in. A buffer is a solution which consists of a weak acid and its conjugate base that can resist a change in pH when a stronger acid or base is added. All right. Well, I'm sure we know we have a little bit of an idea of what buffer systems are, what acids are, bases are, bufferings are. We must have done that in chemistry classes before this stage. All right. So the bicarbonate buffer system is the most important extracellular fluid buffer system. All right, and the buffer pair consists of bicarbonate and carbonic acid. All right. All right, so bicarbonate is formed in the red blood cell, catalyzed by carbonic anhydrase, as discussed earlier. Then we also have the hemoglobin buffer system, which is a protein buffer system, and this is the most important non-bicarbonate buffer system of blood. All right, so these are the four main functions of blood. All right, so with that, we have come to the end of the lecture. Thank you for listening. I will see you later. I expect you to make note of any questions you have or any clarifications you might require. This will all be answered when we meet at our next class. Thank you.